Okay, everyone, good morning and good afternoon, wherever you are. Thanks for joining us today for another fine seminar. My name is Lauren Hayes. I'm your host today. And as usual, we're looking forward to another good talk today. Last week, we had to cancel. Hopefully our speaker's faring better, was not feeling well. But next week, I just want to announce that we have Trinity Bilde from Aarhus University. She'll be talking about social spiders. And for today's seminar, it's the usual format. We have about 45 minutes of seminar following discussion. We ask that, please stick around for the discussion afterwards. As usual, we'll be doing in the strategy that people post questions to the question mark to the chat, and then I'll relay that to the speaker today. So this brings me great pleasure to introduce today's speaker. It's Luis Ebensperger from Chile. Luis is a professor at Universidad Católica de Chile in Santiago de Chile. He earned his PhD at Boston University under the advice of Robert Tamarin. He then did a postdoc at the Universidad Católica de Chile with Pancho Bazanovic, a well-known physiologist. And he has research affiliations in various parts of the world, including the University of Munster in Germany. Luis's research, and I can speak from some knowledge, Luis and I have been collaborating together for many years now. It seems, I think it's 15 or 16 years. A remarkably insightful individual. He sees the big picture and asks really interesting questions. He uses a combination of field work, lab studies. He's done some comparative work to understand why animals live in groups, why they cooperate, and also some of the mechanisms underlying those behaviors. He has also generated a lot of interesting data on social instability, which you'll hear a little bit about today. And much of his work, he has worked on a number of species ranging from voles to some of the South American caviomorph rodents, but his primary study organism is the degu, Octodon degus. And you'll hear a lot about degus today in part of the long-term study that he's been working on. He's also edited 2 books, one of which is a book on sociality and caviomorph rodents, and has also recently edited a book on social living in the native fauna of Chile. Luis has been a prolific publisher. He has almost 100 publications in peer-reviewed journals, ranging from journal Animal Ecology, Brain Behavior and Evolution, Hormones and Behavior, all the big behavior journals Luis has published in them. So it shows the kind of the range and the integrative framework that he's been developing through his science. He's got considerable funding. I think I counted 7 FONDECYT awards, which are the equivalent in the US to a National Science Foundation grant. So been keeping the project going for many years now. And he also has been recently awarded by the Animal Behavior Society the Quest Award, which is one of the highest honors you can get from that society for scientific achievement. And I can say that he's a great collaborator. He's worked with numerous grad students, undergrads, and he's helped tutor my students in Chile. I spend some time and I leave them there. And Luis has been very patient with me and worked with over 40 students of mine from the United States over the years. And so he's a great colleague. And so today, Luis is going to be giving a talk entitled A Model of Sociality Driven by Instability, Sex Conflict, and Context-Dependent Benefits. And with that, I'll turn it over, Luis.
Okay. Thank you very much, Warren. Okay. We'll get this. Okay. Hope that is the
Looks
right
good.
screen here.
Looks good, Luis.
Thanks. Oh, sorry. Okay, well, thank you very much to all for coming here to listen this talk. I'm grateful to the organizer for all the organizers for of this international series seminar for the opportunity to be here and communicate some of our findings that that have emerged from a highly collaborative research I have been a part of. So today I would like to focus on what we have learned about social organization and cooperative behavior of the degu, mostly agave rodent. So I decided to do this through contrasting, putting this in the context in which I will contrast attributes of degu sociality with what we might expect for average group living species, if you want. Ultimately, by doing so, I wish to highlight how social behavior of these animals somehow extends our understanding of mammalian sociality in particular. So here, sorry, here are the main points that I would like to address. First, I will show or highlight some selected characteristics that we generally associate with group living species. Second, I will address some basic aspects of degu ecology and life history that are relevant in this context in particular. And thirdly, I will describe some main attributes of degu social organization, mating system, and some of cooperative behavior. Finally, in the final part of the talk, I will refer to what we know about fitness consequences emerging from these aspects of degu social organization, mating system, and some potential mechanisms that might be involved. So that's hopefully that will be not too much. So I will begin by saying that whenever we think about group living species overall, we normally imply that we are dealing with individuals that live in generally well-defined groups, and meaning here that adult group composition generally is stable, while it may be conditioned, of course, by predictable events of reproductive emigration and immigration, and of course, some mortality. Additionally, and especially in species in which unimale, multimale, and multimale, multi-female groups are common, mating interactions generally involve same group members. So even some variation, of course, exists. Social groups tend to function as reproductive units. Regarding the recruitment of new members, it occurs typically, but not exclusively, that this process includes the retention of offspring of the most philopatric sex. So there's a high potential in several species for the formation of kin-based groups. On the other hand, if we look at now the aspect of social structure, affiliation interactions, also are relatively more prevalent among same group members, which may lead to the formation of bonding relationships, as it has been reported in several highly social mammals. Eventually, these affiliative bonds contribute to the establishment or to different forms of cooperation that we observe, and where the associated fitness effects of these cooperative relationships may typically are population specific. An additional point that I would like to highlight here, because of it's very relevant to what I will be addressing later, is that studies on, especially studies on social birds and mammals, indicate that permanent changes in individual group membership typically reduces the propensity for cooperation in different contexts. In this slide, actually, you see a few studies conducted on cooperative displays and food sharing and exchange. So In addition to reducing the propensity for cooperation, additional studies have shown that these actually also decreased fitness, different fitness measures such as reproductive success. I note here, however, that links between social instability, propensity to cooperate, and fitness outputs of these, in most of these cases, remain little examined within the same species. So here's a main picture of the modeled species that I will be referring to today. Before I address how social degus fit to the previous generalizations, I would like to introduce a few attributes of the degu ecology and life history that are relevant here. So regarding some basic ecology, these are you now can see represented representative images of 3 different populations that we have examined to a different extent. On the right, on the left is a main picture of what used to be our main study site, and they show that the preferred habitat used by degus is generally open or exposed in terms of shrub or tree cover, and that these habitats were used to be associated with Mediterranean climate, meaning that we have a combination of rainy and cold winters and dry and hot summers. Additional details about basic ecology in these guys. They was built and used underground burrows to hide. But they're herbivores, and they feed essentially feed above ground from grasses and shrubs. The activity of these guys is strictly diurnal, meaning that these guys during nighttime they just take refuge and stay underground in their burrows. And this is critical because we have been using the patterns of with which these animals share these burrows during nighttime to establish the associations and more directly the size of the different groups that we have been able to identify. Some critical aspects of life history, I would like to show those in the next couple of slides. So here you can see here the typical breeding schedule in our main study population. So these rodents are seasonal breeders, meaning that mating takes place here mostly around June. So it's the transition from autumn to winter. After that, and after a 3 months pregnancy, actually, offspring are born during early September, typically, and the females wean their offspring through October, and dispersal of juveniles begin by late in the spring and early into the summer. Litter size of these animals average 3.5 offspring, so something between 3 and 4. And there's one main breeding event usually. However, it might be we have recorded a second smaller litter produced during some years. That is very the occurrence of these second litters. Typically, we strongly suspect that it's associated to the amount of rain that is produced. So usually when we used to have rainier years, we had a more likely, well, we have a higher probability of this that in our population these guys produce a second litter. This is a very important aspect that I would like to highlight here. So if we look at the adults that survive to next year, we have we recorded the astonishing view here that inter-year survival of adults is generally low. You can see here, if I can use the pointer, So in this figure, you have adult survival. So here, so all in this set so far, you have the proportion of adults that were alive during the breeding season in our winter, that survived to spring of the same year, summer of the same year, and next winter, that is the coming year. So it means that about 10 to 15% of the breeding adults that are alive to mate during one particular winter make it again to breed during the consecutive year. So it's a very low proportion of individuals. We have suggested that degus are essentially, because of this, degus are essentially ecologically semmelferous. Now I will address some of the attributes that are frequently observed when it comes to group living in this species. This slide is to just for you to appreciate the regular protocols we have been followed to monitor social organization in these animals. So we basically use night telemetry, live capture, and recapture during very early morning hours. We use this information to establish burrow sharing in the field. And then from that information, we quantify the size and individual composition of such groups. As most of you may be aware, every time a new animal is captured, we take some small tissue samples and we use that to determine maternity, paternity, and relatedness, and eventually estimate reproductive success. Since 2009, I think this is important for you to follow and understand here. Since 2009, we added we conducted these kind of assessments to determine social organization twice per year. One during one instance overlapping the main mating time of the year, and the second one the time overlapping most of the lactation. So we actually use then information about the size and individual of group composition of these guys in such groups to determine potential and eventual changes in their individual group composition. So here you can see I'll now show you some general a few pictures that illustrate some general features of the social organization. So, and in this one, it shows that the social organization in Daegus is relatively diverse in terms of group sizes. So here you can see on these two figures the different frequency with which we have observed different size groups on the left, and groups with different numbers of females. That is shown here on the right. So on average, mean group size in Daegus in our main study population is about 3 adults, and out of these, 2 are female members. In this other one, here, this figure illustrates that social organization in these animals is also diverse in terms of adult sex composition. So, so here on this axis, you can see the different type of social groupings that we have been able to record over the years, and see that you can see hopefully that the most common social units include multiple females without males, multiple females with 1 male, or multiple females with multiple males. We have observed also secondarily females that nest solitarily, as you can see here, and also female-male pairs. Rarely, however, we have recorded all male groups and solitary nesting males, as that is, you can see here in the figure. An additional interesting characteristic of degus, I would say, is suggested here in this figure. So degus are also colonial, meaning that main burrow systems used by different groups may be spatially clamped. And that is, to help your following here, you can see that here I highlighted the main burrow systems used by neighboring groups. What about the stability of the social organization? Well, our data on individual group composition during mating and lactation revealed that this social organization is generally fairly unstable. So here you can see a main figure in which out of 99 groups that we were able to monitor through almost 11 years, 85% of these groups, there was evidence in these groups that they have experienced one or more changes in individual group membership. And on the figure, on this bar chart on the right, you see the origin associated to these changes in group composition. And most of it has to do with the movement of adults in and out of the groups, and to a lesser extent by natural mortality, mostly because of predation. Only a few, a minority of the social groups we have recorded, that is 13 of them, were actually completely stable during the main breeding season. This figure complements the previous one. So here, if we focus at the level of the degu, you can see that on average per degu, each group member experiences about 2.5, between 2 and 3 changes that were linked to immigration or emigration, or the movement of individuals between groups. I would say that pending an update of this is a relatively old paper already, but because despite that, we also reported that social groups exhibit a relatively high turnover in adult group membership when we focus across years. That is probably not that surprising given the information that I just presented to you before, in that degus exhibit a very short lifespan across years. So here you could see that if we look at the origin of group members and where if we establish here, we try to establish the origin of group members, and when we look at the group composition during any particular year, we see that most members in those groups are represented by female and male adults that were immigrants, and secondarily by female members from previous years, and that is shown probably here. You can see the proportion of this here, and also by some philopatric female offspring of the same group, that is shown here, and here. We also have as group members some male and female offspring of members of other previous groups. So essentially, most groups experience an exchange in terms of group membership, and usually one group hardly continues to the following year if we follow the individual individual group members. Okay, what about the mating system? We published this analysis relatively recently. We show here that the mating system of degus is both sexually and socially promiscuous. And what I mean by this is that most males and females produce offspring with multiple opposite sex partners who include members of the same or neighboring groups. So the implication here is that social groups are not necessarily closed units when it comes to mating interactions. What about kinship in terms of social organization of these guys? So far, we have conducted 2 examinations based on population genetics, and both of which consistently reveal that within group kinship is actually not different from random expectations throughout the population, suggesting no kin structure in at least 2 populations we have examined thus far. So therefore, individual group membership it tends to be non-kin biased. What do we know about cooperative interactions? Here, I just would like to summarize. We have conducted several experimental studies in the laboratory and other ones in the field that suggest that degus exhibit a variety of different cooperative interactions or in different contexts. And some of these include the communal care of the offspring, social vigilance and enhanced predator detection during foraging, and also during the digging of the burrows. They actually can we know that these guys may actually coordinate their digging when they do this. So I would like now to go into the final point and see how fitness and revise here the fitness consequences and some of the potential mechanisms that might be involved after showing you the previous information. So my tenet here is that degus actually exhibit a relatively unusual social organization characterized by instability in group membership overall, by social groups that do not constitute closed units in terms of mating interactions, and however, they show frequent cooperation among group members that are generally non-kin. So in this last section, I aim to determine how these attributes actually impact fitness in this and the mechanisms that may be involved. Here, this is also relatively recent information. Here we reported with regard to the mating associations or the mating interactions. We reported that multiple mating is actually common in both males and females. However, our examination revealed that this sexual strategy benefits the male only. And that is shown here, where you can see that the males, as the number of the males mate with a larger number of group mates, or mates from neighboring groups, they increase the total number of offspring size. This is, of course, not the case for the females. In this other one, in this other study that is also correlational in nature, we examined. I would like to show an examination of the fitness effect or the fitness consequences associated with group membership instability. So in these figures, I would like you to take a look at this. What these figures show is how the number of offspring weaned by females on the left and males on the right vary as a function of instability in female group composition. And this goes from total stability over here to highly unstable group here, and the number of females on these other axes. So visually and also an analytical, of course, we recorded that females win more offspring when in groups with more socially stable females. On the contrary, males sire more offspring when in groups with more, but socially unstable females. The same was true when we actually consider instability in male group composition rather than female group composition. So essentially, the results are pretty much the same. We got that females win more offspring when in groups with more females, but when group males are relatively socially stable. In contrast, males sire more offspring, and this was associated with groups with more females, but also when in these groups, group males were more socially unstable. So the overall picture was here is that females benefit mostly whenever group membership is relatively more stable, but males benefit the most under socially unstable conditions. I would like to also mention this subsequent study. This was led by my colleague Lauren Hayes, and it adds an additional point to the big picture here, and although not necessarily to the indirect connection with stability. So here we look at is how the presence of one or more males within multiple female groups actually had any consequence on the females, and they actually did. Again, this is a correlational based study. We noted the associations here that suggest that the presence of one or more males in these groups actually decreases the mean number of offspring produced by each female within groups. Not only that, but they seem to, this effect is also associated with an increase in the variance of the number of offspring of the females within groups. So females when females experience males as group members, they tend to produce less offspring, and the production of offspring among the females within groups becomes more unequal. Okay, so now I hope I will revise a few, I will show you findings from a few studies conducted under laboratory conditions that in which we have aimed to try to unravel some of the potential mechanisms that might be involved in this fitness patterns that I just described to you. So in this one here, we exposed, I would like to go slowly so you can, hopefully you can follow this. So we expose groups of one breeding and two non-breeding females to two events of group instability by replacing one of the socially familiar non-breeding cage mates each time. And then we compare the care by the breeding females and the adult care exhibited by the non-breeding females in socially unstable versus socially stable groups. So the figures show these two main conditions here, stable groups and unstable groups. And the responses that are here are the time spent by these adults with the offspring and the number of active parental interactions received by the leaders from these adults. So the black bars here are for the breeding females or the mother, and the gray bars are for the behavior of the the non-breeding females. I think I should point out that with the exception of lactation, of course, the quantity and the quality of care exhibited by non-breeding females in degus is essentially similar, almost identical to that exhibited by breeding females. So a main result that we got here is that compared with socially stable groups here and here, non-breeding females in socially unstable groups tend to decrease adult care to the offspring of the breeding female. So you can see the the larger differences between these two and here too. And the breeding females in turn tend to increase their own maternal behavior as an attempt to compensate that. This other figure, it's one of the main results of a subsequent experiment. And here, what we did was this was a 2-year experiment in which we quantified how stability of social conditions experienced during development by the females influence their own social and maternal behavior of these after experiencing either stable or unstable pregnancies. So we followed these females from the time in which they were in their early development and then as breeding adults. So here you can see on this figure, we have one of the main effects recorded. So here we have the socially stable females that experience social stability during their early development. And these are the females that experience social instability during their early development. So the main result here is that females who experience social instability during development, as suggested by these differences, enhance care of their own offspring, regardless of stability conditions experienced later as breeding adults. There was a little surprising in terms that this suggests a potentially unflexible strategy by the female to shape the phenotype of their offspring to potentially face socially unstable conditions. Yet, however, this seems consistent also with the observation, is nonetheless consistent with the additional observation that social stability experienced by degu offspring under natural conditions during 1 year does not predict actually stability to be experienced by the same individuals as breeding adults during the subsequent year. We have also published this information in a different paper. Finally, with regard to potential mechanisms involved here, I would like to just show some of the main findings of this additional study. Here, in this study, we examine the contribution of the male sires to the offspring care and his effects on the condition of breeding females. So we compare the maternal behavior of and body condition of breeding females that were nesting solitarily, and you can see this condition on the left, with that of the breeding female nesting with another non-breeding female here that was socially familiar with her, and finally with a condition in which a breeding female stayed with the male sire. So from here we allow the females to give birth to the breeding females to give birth to their offspring, and we examine maternal, paternal, and alloparental care, allocare, body mass of adults and offspring, and baseline plasma cortisol. So the main results that I would like to bring here today are shown in these two figures. Here you can see the loss of body mass by the breeding females in these 3 different conditions, and also the plasma cord estimates obtained from breeding females in black bars, and also for the non-breeding female in the case of the in this social condition, and for the sire in the 3rd condition here. So the breeding females actually experience positive effects in terms of losing less body weight and show lower baseline levels of plasma cortisol when nesting with a non-breeding female. So you can see here that under this condition, the females, the condition of the females got better as they lactate their offspring. In the figure, in the down figure here, you see that how the plasma cortisol values are smaller, significantly smaller for the breeding females, only in the condition in which they were allowed to stay with a female partner, with a non-breeding female partner. So in this way, and into the context into the talk, the presence of the males seems generally detrimental to the body condition of the breeding females, even if the male is actually the sire of the offspring. So my overall view is that currently Daegus represent a rather unusual model of sociality, in which different forms of cooperation coexist within a social organization characterized by frequent and permanent changes, and that is sexually open, where social benefits are sex and social condition specific. And here I use these figures to summarize the female and the male side of this story. So on this side here, you can see that in general, we got evidence suggesting that female group members contribute positively to the female's fitness and through the communal care of the offspring. And the one critical addition here is that instability in group membership is potentially acting as a modulator of this. And most of the time, actually instability in group membership interferes with communal care, generating a cost to the females. Also, we also have previous evidence suggesting that the presence of the males within groups is generally detrimental to the female condition, and also in terms of the number of offspring produced. On the other hand, the male side, males thus far, well, I would say we have been studying them with less detail. Males seem to benefit the most here from living in multi-female groups, and where instability in group membership actually seems beneficial to them. And we strongly suspect that it's likely has to do with an enhancement of mating opportunities, driven mostly by instability in group membership. We also may consider that an additional effect here in terms of increasing mating opportunity has to do with also with the colonial nature of Daegus overall in their environment. Okay, I think I don't know how. Okay, I'm almost done. I just would like to finish here by saying or showing mentioning a few additional directions we are currently aimed to examine or explore. And the first one I would like to mention here is that recently a colleague of ours, Loreto Correa, reported that females in the population in which we have been studying Daegus are sorted positively in terms of anogenital distance, which is an important correlate of in utero masculinization during development in Daegus. So females that tend to be more masculinized in the population tend to associate and be members or share group membership with other females that are also more masculinized. Not only that, we also determined that when we examine the fitness correlates of this, we determined that litter size of females increase with mean AGD of same group females. So in other words, more groups that include more masculinized females tend to produce a larger number of offspring compared with females in groups with more feminized females. Now, since AGD, why this is important for the previous context? AGD is associated with variation in different aspects of social behavior. So we aim to determine how relatively more masculinized females are better to either prevent social instability in group membership, or and for instance to exhibiting greater patterns of affiliation. Additionally, we are heading to examine a scenario in which social instability may actually represent an opportunity to some extent to the females as well, in terms that by experiencing instability, females may actually attempt to rearrange their social association through forming either homo or heterophilic groups in terms of behavior. I think you may remind that, and I should that social instability involves not only male movement but also the movement or immigration of some of the females too. So I guess I will now end this, and I just would like to say thanks to everyone for listening, and I'll be happy to address any questions.
Thank you, Luis. Of course, I found that fascinating, interesting. So this is a chance for us to have questions. Again, please remember, please post your question marks in the chat, and I'll relay those to Luis. And since we're trying to build a community here, please tell us, who you are, where you're located, and really try to encourage students to ask questions, or junior folks to ask questions in this format. If you're in YouTube, please just post your full question, and I'll relay that to Luis. All right, so we'll get started. We have a question from Eduardo.
Sure.
Thank you, Lauren. Eduardo Fernández Duque, Yale University. Luis, thank you for a very, very interesting talk. I'm hoping that something will take me to Chile to visit you and see the field sites. I have rather concrete questions that speak to understanding the system a little better. How much the degus weigh on average? And then you were, if I remember well, you said that the pregnancy is about 3 months, which struck me as a rather long pregnancy period, given what I'm assuming their weight is. And finally, just for you, if you can comment on some life history aspects. If they're dying, pretty much all of them, within a year, do we have estimates of lifespan in captivity? I'm assuming that they are dying because of primarily extrinsic factors of mortality. I'm just curious, what would be the free-running mortality or lifespan if they were pampered in captivity?
Okay.
So kind of very
okay.
factual.
I'll try to remember. Okay. Please. I mean, just remind me again if I'm losing some of the points that you addressed. You asked me. So these guys, I mean, the average body weight of these guys is about 180 to 200 grams animal. So it's I would say it's a rat size, not big rat, but a medium sized rat overall. Now, then you okay. So
pregnancy length.
Oh, okay. This, I mean, the pregnancy is actually is a very unusual life history trait for a rodent actually, and so if you compare with and the reason for that obviously is that offspring, when they are born, are very mature, and they're completely furred. And they, in the case of the degus, they are born with their eyes open. So they are very mature in terms of their neural system, And so they just very ready to move around easily. I mean, even though they're not extremely mobile during the first 10 days, but they rapidly mature in terms of moving around. What else you were interested?
And then I'm just curious, for their lifespan, they have very short lives. Is
okay.
that
Yeah.
primarily exclusively extrinsic factors of mortality? I mean, would they live 10 years if you had them in captivity?
Okay, yeah, I'll try to be very straightforward here. In our study population, adult survival is that short, as I show you. We have evidence from a different northern population where degus may probably live 2 years. Now, in our population, we have some cases, I would say about, I mean, it's a northern in the same amount that I already told you. So 10 to 15% of the cases, the females and some males may actually attain to a second or even sometimes to a third year. But that is unusual. It's very unusual. And so overall in that population, most degus by far live only one year or one breeding event usually. Probably they live chronologically. They overpass one year if you consider. I mean, the more or less the month in which these animals are born. However, that's the rule. That is in striking. There is in striking contrast with what we know from degus in captivity. I mean, most of you, the ones that are in Europe and the US, are familiar with degus because, maybe familiar with degus because they are pets there. And also they are used in biomedical studies. So under laboratory conditions, these guys may easily reach 3, 4, or even 5 years. I have a colleague here, that was a former graduate student of mine that has a degu colony, and the animals easily reach 4 to 5 years. And that's the reason why, maybe I may use this to answer your question, because these older degus in that you can have in captivity, they exhibit symptoms of degenerative diseases that affect humans, like Parkinson's disease and others, like Alzheimer. So, and that's the reason why degus are becoming a popular model in these other area of research. So my view here is that, ecologically speaking, what we are doing is with the degus is that we are preventing them to. We are taking degus when we bring them to the laboratory. What we are doing is getting rid of any natural selection forces, and these guys actually live longer in the laboratory conditions. So my view is that these guys, I mean, they're mostly the mortality they suffer has to do with ecological factors, and probably with the tire and wearing of living in harsh environments, where food is sometimes, very low quantities in their natural environment.
Thank you very much. Thanks a lot.
I missed something. I don't know.
No. I learned a lot about the natural history of the degus. Thank you so much for a wonderful talk.
Sure.
Okay, so our next question is from Michael Taborski.
Hi,
Michael Taborski, University of Bern, Switzerland. Hi, Luis. Very nice talk and interesting study system, really. I just would like to understand a little bit better the function or the role of these non-reproductive females? I mean, given that there is such little survival chances between years, and these animals are apparently very seasonal, how do you explain non-reproductive females at all?
Oh, okay. I did it. Okay, I think I apologize, but I didn't address what I understand your question, and it's an obvious question without the information I will give you now. Normally, most females in the population breed. So essentially, we have been using non-breeding females as an instrumental, simply instrumental to our experiments, because they still exhibit allocare to the offspring of other females. And what we normally face when we try to run these experiments in the laboratory, in these artificial social groups, we usually have it's very difficult to synchronize females so that they can actually breed at the same time. That's the main reason why we ran most of at least 2 experiments thus far using non-breeding females instead of just simply multiple breeding females.
Okay,
however,
in nature they would be communal breeders then.
Yeah, they in nature they will be communally breeding their offspring.
Okay, and then also, do you know whether they would be allosuckling each other's offspring?
Yes, we have observed that, but only in captivity. We have witnessed that in captivity, and so they do that.
May I just perhaps ask one last question regarding the group composition, because it's related to this question that I just asked, and that concerns the presence of males. Apparently, I mean, you have quite a few data sets showing that males, or the presence of males in groups, seems to be detrimental to female reproductive success and also survival. So why are there, I mean, do females have any way to control the membership of males in the groups, or are males more dominant and can impose their presence on the group, or how does that work?
That's a central question. We have no answer yet. What I can tell you is that based on the spatial data that we have been collecting in the field, it seems that females are unable to control that males join the group, and so that I don't think that is taking place. So my view, my feeling is that potentially we may be missing some more, let's say, cryptic ways in which females may potentially get rid of the males. I mean, when our social groups imply that the females and the males are sharing more or less, I would say, 2 to 3 meters in diameter burrow system. And these burrow systems inside, they have a main central chamber that these animals where these animals get together. But things that we haven't able to pinpoint very precisely yet is the extent to which females actually do not always stay huddling together with the males at some point. We know that males use the same burrows, and whenever I mean, they do that, they at least potentially can conflict with the females to some extent. But we are not certain about what other more fine-tuning strategies females may be using to try to avoid this costly male.
Okay, thank you very much.
All right, so our next question is Sarah Corsetti, and I apologize.
No.
I don't remember.
Okay.
The name.
Okay, my name is, I didn't succeed in changing the name, I will do.
Oh,
My name is Evgenia Anatoli, I'm in Italy. And so thank you for your interesting talk. But I have to pose a very basic question, important for me to understand, because maybe I missed it. But given that the picture you gave is that there are in small spaces a lot of individuals who made also, you said, neighboring groups mixed, there is promiscuity, etc. What is the criteria to define who belongs to which group? I mean, how can you say that it's 1, 2, 3, or 4 groups? Or
okay. It's like that's an obvious an obvious relevant question. Despite this neighborhood that I show you, our resolution to monitor the spatial associations for these guys is fine enough to distinguish when 2 or more individuals are actually sharing, frequently sharing the same burrow system below ground.
okay.
So essentially, our criteria considers spatial associations during the nighttime, meaning the time in which these animals are not active above ground, but they are presumably resting or being underground, and closely associating between them.
Okay. Thank you.
Sure.
Okay, our next question is from Maren.
Hello, my name is Maren Hoek. I'm from the University of Derby in the UK. And thanks very much for the very interesting talks. And I have lots of questions, but I try to refrain myself. So one is a bit of a follow-up question on the natural history to things that Eduardo also asked. So I'm really intrigued by this fact that they have so few offspring, considering that they have only, essentially the females only 1 litter. I mean, if you compare it to guinea pigs, who also have a sort of case strategy for rodent, but at least they have lots of litters. And they have 3.5 offspring on average, but only a single litter. So the survival rate of the offspring must be really high then. Is that true? Or I mean, relatively high for such a small rodent.
Yeah, it's about 50% or 60%, pretty much. So meaning that the offspring, I mean, that is the percentage of the offspring that you will see in the same population next year.
And that's what you would expect given the numbers that you gave. Otherwise, they would go extinct if you have a lot.
Yeah, right. Exactly. But also notice that, I mean, we pretty much in our long-term study population, we have been doing extensive trapping and extensive trapping effort. However, we cannot fully rule out that offspring from other groups in the neighboring area may actually also move in the population. So it is not that simple by saying that 50% of our offspring are enough to keep the population going on in this case. So we may have some immigration during some years.
And sort of following up on that, if they have, you describe them as sexually promiscuous. For the females, I mean, for the males, okay, they can mate with several females, but if a female has only 1 litter, did you look at paternity? Do you have lots of mixed paternities, so several fathers in the same litter?
Yeah. That information was on the slide, but I probably use it. So let's get to it. Yeah, so essentially our data indicates that about up 80% of the males actually and 65% of the females produce offspring with 2 or more opposite sex individuals within groups. However, most males, 77% of the males and also 64% of the females actually produce offspring with that include individuals, males from the neighboring groups. So in a sense, we have a figure here that most leaders of every female in the population almost include multiple paternity. And that multiple paternity may come from within group males, but also from neighboring groups.
Okay, thank you very much. I posted another question mark so others can come first and then I ask more questions if there's time.
Sure. Thank you.
Thanks, Maren. Next is Leticia.
Hi, Lauren. I think you skipped Adriana before me.
Adriana lost connection, she told me. So you're okay.
Okay, so I'll go ahead and Adriana come back. Hi, Luis. Really nice talk. Thank you so much.
Thank you.
Yeah, so I have a follow-up on the multiple female mating. So you said that males benefit by producing more offspring siring more offspring when they mate with multiple females, but females don't. But could they have a benefit in terms of having more genetically diverse offspring when they mate with multiple males? I don't know whether that could be figured out empirically or not, but what is your take on that?
We did not in this same study. We reported that genetic variation associated was not a good predictor of multiple mating in the population. Actually, there was no association with that. Mm-hmm. So I mean, yeah, there obviously, if I remember correctly, there might be several other potential explanations that are by which females may get some more cryptic benefits associated, but we haven't gone through that. Thus far, we only we focus mostly on the basic aspect of whether there was any association between litter size of the females and having all these different sexual partners during the main breeding season.
And may I ask another question, kind of separate? All right.
Sure.
Yeah. So I'm curious about. So what do you think is the ultimate driver of why they live in groups? Could it be that it's just limited space and they just kind of share burrows because, there isn't any other place to go? Or is there some intrinsic benefit? And I guess you kind of, and that intrinsic benefit, if there is, what would it come from? And it seems that it could be from protection from predation by kind of these vigilance and maybe the communal root care within the burrow. So my guess, a way of testing that would be if there was unlimited space, would they still be forming groups?
Yes. The question is that in general, even though degu density, obviously degu density in the population goes up and down, and they typically remain forming groups. Sometimes the group size decreases or increases, and what we have observed is that whenever density, for instance, is reduced during one particular year, the size of the groups tend to be smaller. So we have observed a density effect of that. However, this is, I think we did some previous examinations we didn't have any evidence to suggest that they was grew because the habitat is actually saturated. So it's really not. It doesn't have to do with that these animals are very they do not have refuge, or refuge is very limited in the population. We conduct some additional analysis to check this. So I don't think it has to do with habitat limitation, in my view. So yeah, predation risk, it's probably one potentially contributing factor to explain I would say I'm not sure about the current sociality of these guys, but I mean, comparative method approaches indicate that predation has been important overall in most caviomorph rodents, actually. I don't know if I follow your
yes,
question.
yeah.
If I missed something, please let me know.
Yeah, unfortunately, my internet dropped for a second there, so I missed part of your answer. I think I get the gist of it. That
sure.
Yeah,
because if not,
probably at least historically the main driver, right?
Yeah, I guess so.
Yeah, thank you. No, that definitely answers my question. And then there is no habitat saturation also. Yeah, thank you.
Sure, thank you.
Okay, so our next question is from Zulema.
Yeah, hi, Zulema Tang Martinez, retired from the University of Missouri in St. Louis, which is right in the middle of the United States. Thank you, Luis. It was a really interesting talk, and it was nice to see so much data sort of all brought together in one place. So that I really like that. I have two questions. The first question is, towards the end of your talk, when you talked about fitness consequences for females and the effects of having disruptions and having a non-breeding female along with the breeding female, I wonder if it makes any difference in your results if that non-breeding female is genetically related to the breeding female, and then I'll ask my second question afterwards.
Okay. No, we did not did in that experiment. So I mean, the females the females in those experiments were not sisters. I mean, they are not closely genetically related. Okay. We did this because previous experiments during the first decade of the 2000s, I conducted additional research with this type of things, and I mean, different lines of evidence, both in the lab and in the field, suggest that relatedness does not affect communal care in the female degu. There's one published paper, actually, in animal behavior that suggested that that was conducted with degus from the States, and that suggested that mothers may distinguish, or I mean, they can make a difference between own and different offspring when communally nesting their offspring. We did not observe anything of that.
Yeah. Okay, thanks. The second question is, given that there seems to be a fair amount of mating among members of different groups, in addition to the mating, how much interaction is there among the different groups within a colony? And I guess that the bottom line question that I'm asking is, do you think it's possible that the colony itself could be the basic group that is then subdivided into smaller groups? And I'm thinking of like capybaras, where you can have these huge groups, but they're fission fusion, and so you at any point in time you can find little groups, subgroups that for days and weeks, wander around together, but in reality, they're part of that larger group.
Yeah. Okay. I guess I'll try to organize. So I see the. I mean, my view is that what we see in Daegus is not a fusion society. When you, I mean, in this case, compared with you make the contrast with the capybaras. So what we really, in periodic, what we consistently record here is that individuals keep close associations in terms of sharing their burrows with some individuals. And when they switch groups, they switch permanently. So that is a main difference to me, in that we never have observed. I mean, I don't have records of individuals that switch group and then come back to the previous association. Okay, I may be missing maybe one or a few cases, but we don't see that. And I think that makes Daegus a little different from I would say a typical fishing fusion society. Now, regarding the interactions that we normally see, we have conducted, at different periods during our studies, we have watched the animals directly in their environments and record social interactions. And what we can see is that most of these involve individuals coming from surrounding areas and fighting and being really agonistic with individuals that are located in neighboring burrow systems. So again, this suggests to me that it's not a huge a colony that works like a single main group. Again, so I think it's a very, I would say, I don't know, midterm situation here, in which we have particular groups that are, packed together spatially, but that in some contexts, for instance, during mating, you see chaos. So individuals actually interact sexually with individuals from different small groups.
Yeah, in some ways it sounds more like the prairie dog, the black-tailed prairie dog situation, where you have a town and then you have very distinct groups within that town.
Right.
Yeah.
But my understanding that is that individuals are very aggressive towards the individuals of neighboring coteries. I think.
And as far as we know, there's little mating, like between different coteries within the prairie dog town. Yeah. Thanks. That helps a lot. Yeah. Thank you.
Sure.
Okay, our next question comes from Logan.
Okay. Hi, I'm Logan Platt.
Hi, Logan.
I'm in Lauren's lab at the University of Tennessee at Chattanooga. So I was wondering, when looking at different populations and different sites, have you noticed any differences in social organization that could possibly be based on environmental differences between the populations?
Yeah, that's a good question. We haven't. Well, we do have a recent paper suggesting that there's no main differences in social organization between at least two populations in the northern area of Chile and in our population in near Santiago. If I recall correctly, there might be. To be honest, we haven't conducted. Long-term comparisons with those populations, but we have recorded that a few differences in terms of the numbers of group members in Fray Jorge, a northern population, and our population in Rinconada in Santiago. And so in the northern, if I remember correctly, yeah, the population we are studying, group size tends to be a little larger, if I remember correctly. And the other contrast we did is with a population in the Andes. We contrasted our main population with that other population, and we also reported a few differences in terms of total group sizes. I don't think there were important differences in terms of sex composition within groups. So.
Okay, I was just remembering that, like
however,
you said.
However, I mean, something that we don't know is whether actually social structure or how the interactions may actually predominantly differ between the populations.
Okay, yeah, I was just remembering that you said that the northern population sometimes had a higher proportion of the population living to the next year to reproduce. So,
yes, that's an important life history difference with that population.
Yeah, and yeah, I guess that would be mostly based on environmental conditions, like they have more food up north or something. Do you think that's the case?
Well, presumably yes, because that population is very special, is very close to the coastal areas, and so you have much more. I mean, even though you don't have necessarily more rain in here, but you have more humidity because of the fog.
Okay. Oh, thank you.
Okay, we come back to Maren.
Right. During your talk, I first had the question, when you talked about that instability decreases essentially the fitness of the females, that obviously led to a question that you partly answered, which is why are females then still sort of so unstable in their group composition? And then you talked in that context as perhaps part of the explanation about this anogenital distance and the masculinization of the females. And there was the one graph which I didn't find, sorry, but I didn't find particularly convincing, that was the correlation between the anogenital distance or the masculinization and the one on the right hand. And
number of us.
The anogenital distance and the
you mean fitness or the anogenital distance of the partners of the group?
Yeah, so you had on the one slide, you had two graphs. One was about that the females associate associatively, and the other one was about the anogenital distance and I think reproductive success. And,
yes,
and
side.
essentially the confidence intervals were very large, and if you just look at the dots, it was sort of all over the place. And I wonder whether there is, well, I felt that there might, that's probably not quite the full picture to say the more masculinized have higher reproductive success. And I was wondering whether there could be year-to-year differences where perhaps one phenotype has advantages, and in other years it might be something else, and just by chance for the moment you haven't had the years.
Well, I mean, that analysis took into account the potential year effects, and so that pattern is, I mean, took that into consideration. So we didn't find eventually some indication that that relationship may change. I mean, across years. In that case.
But the relationship in itself, I mean, it might have been significant, but there wasn't a strong relationship because you had so much variance all across the conditions that it didn't.
I see. Okay. Yeah. Well, I would have. I mean, it's only as well. I agree. Probably agree with you. I mean, it's a trend, and probably the effect size is not huge. Of course, sure. I agree with that. I added that because it's only to suggest that females, the main point that was that females may be critically doing or using strategy to deal with this instability. And what I really like to check is whether females use instability or this unstable social environment in which they live to associate in different ways to actually improve their own reproductive success. Even though they have to deal with this male instability included in the social organization. So that was only my purpose by showing that.
I mean, I think there's something very intriguing going on there. It sounds very interesting, but I think that probably more research, more questions, yes, more answers. Thank you, yes, very much.
Sure.
There's no more questions in the queue. Does anyone here have a question like to ask Luis? Karsten?
Yeah, it's Carsten Schrading from the CNRS in Strasbourg, France, studying rodents in South Africa. Thank you for a fascinating talk. I have a question that doesn't really have anything to do with the data you showed, but with some of your photos. And it's maybe a stupid question, but something I've been wondering about our rodents for a long time. It's about these runways we see from these burrow systems going everywhere. Do you have any idea how functional these runways really are with going to the best foraging ground, or what defines the geometry of these runways?
Oh, that's an interesting question. I think that is that is an aspect that has not been actually addressed very carefully, I guess. We know, however, I had a graduate student at some point in the past, and she did some research associated with this, and determined that actually runways are important because what they do is use these runways that communicate different burrow systems, but they use these runways to also visit different patches when where they go to foraging. So it's like literally these are like highways. Where they move when they go abroad to get to forage. So what they do is they use the runways, and then from those runways they eventually stop and they take a left or take a right and stay there to forage. I don't know if you sorry, if you ask it something more specific about this.
No, I mean, that's it. I mean, it's kind of what one obviously expects, but I don't know if anyone ever really measured this. I mean, of course. So I was wondering if I would change the runways of my rodents, would they care? Would they go back to the original runways because they're really the best solution? Or how they come into existence?
Well, if you see that, well, the geometry of these runways seem, I mean, they seem to suggest they are, I mean, most of the time I would say that they tend to communicate different burrow systems using relatively straight runways. And you see that alters a little bit in other populations in which you have more shrubs or trees and also big rocks. So in our population, in our main population, these runways tend to be relatively radial and relatively straight overall.
That means it would be more related to getting as fast as possible to the closest shelter, not to get to the best foraging grounds.
Right, exactly. Because as I told you, actually, they look for foraging patches while they go through these runways, and they just go out. I mean, it's not that the runways end on a particular foraging patch. Almost always, these runways end in a particular bird system.
Okay, thank you.
Well, any other questions? I think at this point, well, Eduardo, we'll wrap it up with you because at this point, I know Luis has to teach this afternoon, so we'll do
Then I can
more
follow
question.
up. No, then I can follow up with Luis later. That's okay. I just no problem, Luis. If you're busy, we know that we have more coming up in the afternoon, so we talked about getting together later. I'll just follow up later. Don't worry.
Sure, Eduardo.
I understand.
That will be a pleasure.
Okay, well then what I'm going to do is I'm going to stop the live stream. Before I do, I want to thank Luis again for a great talk and all of you for a wonderful discussion.
Thank you for all.